Hello children, today we are going to start with biomolecules. Biomolecules, what are the objectives of studying this chapter? First, let us quickly have a look at that. So firstly, we'll be talking about what are the characteristics of biomolecules like carbohydrates, proteins and nucleic acids. Then we talk about the classification of carbohydrates, proteins, nucleic acids and vitamins on the basis of their structures. What is the difference between DNA and RNA? And finally, what is the role of biomolecules in the biosystem? So let's begin with the biomolecules. A living system grows, sustains, and reproduces itself. The most amazing thing about a living system is that it is composed of non living atoms and molecules. So, I'll be going through the NCERT textbook also because there are certain points which are very important. And as you all know, that in the examinations we can frame any kind of multiple choice question from the textbook. Furthermore, also we all know that case-based questions are given, so it's very important that we study each and every line. Okay, so especially for the chapter biomolecules, it's very important that I take you through the textbook also. So this is your NCERT textbook. You all must be having this, right? So just let's get back then. So we have a living system grows, sustains, and reproduces itself. So what we are seeing here is that all the living systems are living. They are having a life, but it's very special thing that, or you can say it's very amazing that every living thing is actually made up of non-living things like atoms and molecules. The pursuit of knowledge of what goes on chemically within the living system falls in the domain of biochemistry. So in biochemistry, what are we going to see is what is actually happening inside the living body, like plants and animals. Living systems are made up of various complex biomolecules. What are those biomolecules? Now, first important question now which we have here for the exam is uh, which are the various biomolecules? Now, what are biomolecules? If I talk about is biomolecules are the complex molecules, or you can say the large molecules, which are important for sustenance of life. The meaning of this sustenance of life is that they're very important for us to survive. The examples of biomolecules, as we can see here, carbohydrates, like you're eating chapati, so carbohydrates is nothing but all those monosaccharides, disaccharides and polysaccharides. Proteins, we are all knowing proteins. What are proteins? Like pulses, we have milk, fish, etc. Nucleic acids like DNA, RNA. Lipids, like for example, anything which is going to be fattening, for example, if consumed in large amounts, like oils and fats, are the examples of lipids. Now, they're just talking about briefly about the importance of carbohydrates in this entire paragraph. Proteins and carbohydrates are essential constituents of our food, of course, and these biomolecules interact with each other and constitute the molecular logic of life processes. Right? Then, some simple biomolecules like vitamins and mineral salts also play an important role in the functions of organisms. So I'm sure this is clear to everybody. Like biomolecules are the complex molecules which are important for the sustenance of life, which means they will be responsible for our breathing and other activities. The various biomolecules which are present in the body are, like just now we saw, carbohydrates, proteins, fats, vitamins, and mineral salts also. Today, let's get to the definition of what are carbohydrates. So I think we can see here. Let's change the color to a little better one. Carbohydrates are primarily produced by plants. This is extremely important for all of us to understand. So mostly they'll be produced by plants, and they form a very large group of naturally occurring organic compounds. Some examples of carbohydrates we will be seeing over here are, for example, cane sugar, glucose, starch. Most of the carbohydrates have a general formula CxH2O5. So this was in the earlier times that this definition of carbohydrate was revolving around the formula CxH2O y times. And they were considered as hydrates of carbon. You can see here, this is H2O and this is CX. What does this mean is H2O means hydrate. That is, what you understand what is the meaning of hydrates? It means water. So hydrates of water, carbon. Now, they were considered, this was the old definition. So it's wrong. They were considered as hydrates of carbon, from where the name carbohydrates was derived. So just for the understanding that from where has this word come, it says that they were hydrates of carbon. Example, let's take example of glucose. So glucose is quite important for our board exam also, because it's lots of questions are coming on that. So the formula of glucose is going to be C6H12O6. Glucose fits into this formula, dear children. So if you see that the formula is, like, if I just simplify this, it becomes C6H2O. How many times? If you divide 12 and 6 by 6, then it becomes H2O. That is, this will be 6, right, like this. All right. So the formula will be C6H2O6, the simple formula for glucose. And in this, it indicates that glucose is going to be a hydrate of carbon. But this definition is not sufficient for all the carbohydrates which are existing. There might be some non-carbohydrates which also fit in this definition. All the compounds which fit into this formula may not be classified as carbohydrates. Like, for example, we can say acetic acid is fitting into this formula by simplifying it as C2H2O 2 times. How did we get this formula? We can see that I'll just show you. So if we see acetic acid, it will be CH3COOH, right? So the formula would be actually how many C's you have. You have 2 carbons, right? How many hydrogens you have? You can see there are 4 hydrogens. How many oxygens you have? 2 oxygens, right? So you have to see these two numbers, 4 and 2, and then you can divide these two numbers by a common factor, like let's say 2. So it becomes 2 and this becomes 1, and you divided it by 2. So like this, we can have the formula of acetic acid as C2H2O 2 times, and actually it fits into the carbohydrates definition, that means CxH2O times. But is acetic acid a carbohydrate? If we say, the answer is, it is not a carbohydrate. It is an acid. It's not a carbohydrate. It is vinegar, right? What is acetic acid? It is vinegar. So this definition was actually discarded. Now we can say that it's not a carbohydrate. Similarly, ramnose is a carbohydrate. Its formula is, it's just like glucose, except for the last oxygen you have O5. So C6H2O5, this is not going to fit in the that formula CxH2O times. So we can say that there are certain carbohydrates which are not fitting into that definition. There are certain non-carbohydrates which are fitting into that definition. So this is not good, a very good definition. A large number of their reactions may have shown that they contain specific functional groups. So today, children, let's now see the real definition of carbohydrates. This is important for your examination. The carbohydrates may be defined as optically active polyhydroxyaldehydes or ketones, or the compounds which produce such units on hydrolysis, right? So what does this definition mean? Let's analyze it quickly. So optically active, the compounds actually which can rotate the plane of plane polarized light. So it can rotate, right? So that means the light can rotate either in the clockwise direction or it can rotate in the anti-clockwise direction. You understand this? So the light's direction, if you're passing the light through that compound, the compound, let's say I've taken in a test tube, and this is the way I'm going to pass the plane polarized light. Okay, the light which is just having the vibrations, electromagnetic radiations are just vibrating in one plane. Okay, electromagnetic radiations are just in the one plane, so that's your plane polarized light. So if this light is allowed to pass through this chemical, which is, let's say, rhamnose, for example, you would notice that it rotates the light, the direction of light. So if the light's angle was initially 0 degrees, it will move either towards let's say 10 degrees positive, or it will move towards minus 10 degrees, right? So every optically active compound can rotate the plane polarized light towards the clockwise direction, or that is dextrorotatory compounds, or towards the negative direction, that is anti-clockwise direction, called as levorotatory compounds. So this definition already has been studied by us in class 11th, and also in the chapter haloalkanes and haloarenes, there's a brief mention about what are optically active compounds. We will study that later. So for as of now, this is sufficient for us to understand that optically active compounds, they rotate the plane polarized light, or you can simply say they rotate the light also. If that much also you say, that's quite sufficient. So carbohydrates are optically active polyhydroxy. You can see they should have lots of OH groups in them. At least one they should have, right? They should have the functional group aldehydes, or they should have the functional group of ketones, or the compounds which produce such units on hydrolysis. So some of the carbohydrates are sweet, like chini is there, it is sweet in taste. They will be called sugars. Fructose is sweet, so it's called sugar. And the most common carbohydrate which we are using in our house are like, for example, sucrose, that's the cane sugar, C12, H22O11, right? Now there's another one, children, lactose. It's present in the milk. You must understand that carbohydrates are also called as saccharides. So this word has come from the Greek word saccharon, which means sugar. So from this entire paragraph, as you can see, there are a lot of MCQs which can come up, like what will be is CXH2OY the right definition of carbohydrates? The number 2 is rhamnose, fitting in that definition firstly. So the answer is no. They can give you a variety of carbohydrates, and most of them will fit into this CXH2OY times, but rhamnose is not going to fit into that definition. What are the functional groups present in the carbohydrates? That question can be asked, children. So the best answer is that they contain aldehydes and they contain ketones, as well as just now we discussed, they contain essentially OH, and they all must be optically active. Let us understand what is the classification of carbohydrates. Carbohydrates will be classified on the basis of their behavior towards hydrolysis. When we add water and try to boil the water and adding acid or a base, they break down into small molecules, or sometimes they do not break down. It's not necessary that every time they break down. The first point is monosaccharides. There are 3 types. You can see 3 groups. Monosaccharides cannot be hydrolyzed further. So if you boil them, they are not going to break down. They are the ultimate. That means they cannot be further broken down. So they cannot be hydrolyzed further to give simpler unit of polyhydroxyaldehyde or ketones. About 20 monosaccharides are known to occur in the nature. For example, glucose, fructose, ribose, etc. These 3 you're going to study in this chapter. So what is monosaccharides? Mono means one. So they do not break down further on hydrolysis. Example is glucose, fructose. So at least one line answer should be known by you along with two examples. Oligosaccharides, the word oligo, which means few. So oligosaccharides are the carbohydrates that will give you 2 molecules to 10 molecules of monosaccharides when they are boiled, and they can be further divided into disaccharides if they break to 2 molecules, trisaccharides if they break to give you 3 molecules, tetra means they break to give you 4 molecules, and so on, depending upon how many monosaccharides they are breaking down into. The most common of the oligosaccharides children are disaccharides, which we are going to study in this chapter, right? So here we're going to talk about mostly disaccharides. The two monosaccharide units obtained on hydrolysis of a disaccharide may be same or different. This is extremely important. It should not mean that if I boil a disaccharide, I should get same kind of monosaccharide. I can get different type of monosaccharide. Like for example, in the case of when we talk about, there are three disaccharides which we have to study. We have to study sucrose, we have to study lactose, we have to study maltose, right? So in the case of the disaccharide sucrose, when I boil this, I will get two monosaccharides which will be completely different. So I will get glucose and fructose. I'll get one molecule of glucose and one molecule of fructose. Similarly, when I boil the lactose, I'll be getting one molecule of galactose plus one molecule of glucose. So you can see they are totally different. But when I boil maltose, I'll be getting two same molecules of glucose and glucose. So in this manner, it's understood, I think, that the molecules which are breaking down to give you two simple molecules of monosaccharides are called as disaccharides. These two monosaccharides can be same or different. Now, polysaccharides. Now, in the case of polysaccharides, as you can see, the carbohydrates which yield a large number of monosaccharide units on hydrolysis are called polysaccharides. Examples will be starch, जिसमें हम लोग रोटी बनाते हैं, for example. Then we have cellulose, we have the glycogen, then various gums which are extracted from the trees. The point which is very important here is that polysaccharides are not sweet like monosaccharides. Hence, we can also call them as non-sugars, right? So the first classification of polysaccharides can be depending upon the taste, sweet and non-sweet. Okay, sweet and non-sweet. So the classification of carbohydrates can be on the basis of taste. That means sweet carbohydrates and the non-sweet carbohydrates. So in sweet carbohydrates, children, you can write monosaccharides, and the non-sweet carbohydrates, you can write down polysaccharides. The second classification of carbohydrates can be also on the basis of their chemical behavior. If they are boiled with some, oxidizing agents, then are they able to reduce those oxidizing agents or not? So those carbohydrates which are able to reduce Fehling's solution, that is, and Tollens reagent, are referred to as reducing sugars, and which are unable to reduce, they are called as non-reducing sugars. So all the monosaccharides are going to be reducing sugars, and these monosaccharides can be aldose or ketose. So now here, what is Fehling's solution and what is Tollens solution? This has already been dealt in the other chapters. I'll not go into much detail for the chapter biomolecules. This is important that you just remember these names. So Fehling's solution is actually made up of copper sulfate and alkaline sodium potassium tartarate. So that you will study in the other chapter. So just remember what are reducing sugars. This is important for your board exam. So the carbohydrates which can reduce Fehling's solution and Tollens solution are called as reducing sugars. Example will be your glucose. Glucose is the example, and the monosaccharides, all the monosaccharides. Then all the monosaccharides you can see are reducing sugars. Whereas children, the polysaccharides cannot reduce these reagents. Now, and some disaccharides also are going to be non-reducing in nature. Like for example, we will see that certain monosaccharides are unable, like sucrose, for example. Sucrose is going to be a non-reducing sugar. It is chini, that is sweet in taste, but it is going to be non-reducing. Monosaccharides are further classified on the basis of number of carbon atoms and the functional group present in the those monosaccharides. If a monosaccharide contains an aldehyde group, it's known as aldose. So this definition is important here. Aldose. So what is aldose? Aldose is a kind of sugar. It's a monosaccharide which contains aldehydic functional group, and if it contains a ketonic functional group, children, it's called as ketose. Number of carbon atoms constituting the monosaccharide is also introduced. Now we have some examples. You can see here there are good examples given over here. Let's focus. So if you have 3 carbon atoms, it's called as triose, right? Tetrose, pentose. So in the MCQs, you can have such questions like this. What is the features of a heptose? So the feature of a heptose is that it contains 7 carbon atoms. It's a sugar. It's right. It's going to be a sugar. Secondly, it's going to be called as aldoheptose or ketoheptose. So if it is aldo, means it contains aldehydic functional group. If it is containing ketone, it's called as a ketoheptose. So that's not, I think, very challenging for us to understand. Glucose occurs freely in nature as well as in the combined form. Of where is it found? It's found in the sweet fruits and honey. This is again. Then, for example, all the sweet fruits, children, all the sweet fruits like the ripe grapes, they are also containing glucose in large amounts. So let's quickly see again the definition of carbohydrates. It's written polyhydroxyaldehydes or ketones, or the substances which yield those upon hydrolysis. They must contain at least one chiral carbon atom. So chiral carbon atom, everybody is aware, this is the carbon atom. For example, it should be attached to 4 different atoms or 4 different groups. For example, I can write here, like let's say, let's show you that. So I can have something like this. I can have OH, I can have H, I can have CH3, I can have Cl, for example. So this carbon is going to be your chiral carbon atom, right? The formula which we saw is CxH2O times is not a good formula because all the carbohydrates do not fit into this definition. We have seen the example. Rhamnose is not a, it's a carbohydrate, but it is not fitting into that definition. It's a sugar. They provide us, yes, of course, as I told you, they are important for us. So they provide us food, they provide us clothing, they provide us shelter. Food, like for example the starch. Clothing, for example cotton. There are mistakes in this, some of the spelling mistakes. So they provide us with shelter, like for example wool, right? So wool is also one of the carbohydrates. We have cellulose also, I think, written. So you must understand these points. Okay, let's recap once again. So monosaccharides are the simplest carbohydrates and cannot be hydrolyzed. The formula is CH2O n number of times, right? This is a formula, and this is very simple to remember because it is the form. CH2O stands for formaldehyde, right? The basic formaldehyde, like HCHO. Everybody knows that. So we can write CH2O. So isko yaad, we have to remember this. And so it will be CH2O n number of times. So here we will have n is going to be a number which is standing for 3 to 7. So it can be triose or it can be heptose. Just now we saw that. The examples are the D-glucose, fructose, galactose, ribose, glyceraldehyde. So here, we have taken the examples from the textbook itself because they will be asked in your examination. What is the meaning of D? Let us understand that also. So as we can see that this is the simplest carbohydrate, which is glyceraldehyde, right? So I'll just write the name once again here, glyceraldehyde. So it's very simple to remember. So in this glyceraldehyde, you can see that there are 4 functional groups, different. There are 4 groups, CHO, CH2OH, OH and H. So yes, here this carbon atom is a chiral carbon atom, and you can see that the hydroxyl group is enclosed in a box and it's placed on the right hand side. So whenever you see that the hydroxyl group on the chiral carbon, last chiral carbon atom is written on the right hand side, that kind of sugar will be called as D sugar. Clear? So D sugar means essentially talks about the placement of the OH, the OH on the right hand side. And which OH are we going to talk about? The last chiral carbon atoms OH. Let's see examples. So here we have the example in front of you, the glucose. This is glucose. There are so many carbon atoms in glucose. If you start numbering it right from here, this is the principal functional group aldehyde, so I have to number it as 1. This is going to be a 2, this is going to be 3, this is going to be 4, and this 5, and this is 6. So you can see here that there are functional groups OH everywhere. You can see here there are 1, 2, there's the third one, there's the fourth one, there's the fifth one, children. So total there are 5 functional groups. So which functional group are we going to talk about? As I told you just now, the last chiral carbon atom, the last chiral carbon atom. So this is chiral because this is attached to 4 groups, 1, 2, 3, and this entire thing, right? So this is one chiral carbon atom. Then this is another chiral carbon atom because it is attached to 1, 2. This is the third one, and this is going to be your fourth one. This is the fourth group. So in this manner, this is also going to be your second chiral carbon atom. So in this same manner, if you go on seeing that this is also chiral, this is also chiral. So total, how many chiral carbon atoms are present in the glucose? If we talk about, there are total 4, out of which the last one is this one. So where is the OH in the last one? It is on the right side. So it is going to be called as the D sugar. So the D glucose. And the same thing, if I just reverse it, and if I put it on this side, OH on that side and H on this side, so it becomes your L glucose, right? So there are 2 types of carbohydrates, the D and the L, and all the naturally occurring carbohydrates will have the D configuration, whereas these are the man-mades, the L ones. Okay, so now the second example which we have here is the fructose. So in the drawing over here, you can see that fructose also has got 1, 2, 3, 4, 5, and 6 carbon atoms, and this typically is going to be the last chiral carbon atom, and this OH is on the right side, so that is why it is called as D-fructose. Its name is going to be also called as aldohexose because it's containing aldehydic functional group. It is containing 6 carbon atoms, and it is a sugar. Similarly here it's called ketohexose, right? So I would request everybody who's right now watching this video to please practice this. The basic point which is important for all of us is that we need to practice these structures. So I'll just explain you now how to draw the structures one by one. The basic ones I'll show you first, the glucose and the fructose, right? So you must practice this, children. The formula of glucose and fructose both is the same, that is C6H12O6. So first what do we do here is we draw the Fisher projection formula. On the top you just draw CHO, and at the last one you draw CH2OH, and then you start drawing the H and OH. H on the left, OH on the right. Then you just reverse it, so you just change that pattern. OH and H. Again you change the pattern, so you have H and OH, and you repeat the pattern. So I think this, if you practice, you're going to learn it. Now coming to ketones, fructose, it's also having the same formula. The difference is that glucose contains aldehyde and fructose contains ketone. So of course it's understood that ketone you cannot write on the first carbon atom, right? So you'll have to write this ketone on the second carbon atom. So I'll just change this structure slightly now, children. So this will be your first carbon atom. I'll just write it as CH2OH, and the second carbon atom I'll be writing this as double bond O. So this is the first carbon atom of glucose, CHO. Here you'll write CH2OH. The second carbon atom, we have written C double bond O for the fructose. And the third and the fourth, you should write the same, and the fifth also. So in the third, just copy down that structure as you have done it for the glucose. So you'll have HOH, and you'll have HOH, and on the last one, you write down the CH2OH. So in this manner, you can see that how nicely we have drawn the structure and learned the structure. Then we talked about oligosaccharides. As I told you, oligo means few. So the carbohydrates which upon hydrolysis give 2 to 10 molecules of monosaccharides are called oligosaccharides. Then now let's come to something very important, and this should be learnt by everybody because this is what is going to be asked in the 1 mark questions in the board examination. So the first thing is the classification of disaccharides. We have 3 disaccharides in our syllabus: sucrose, lactose, and maltose. So sucrose is going to be a non-reducing sugar because it does not reduce Fellings or Tollens reagent. Just now we discussed. Lactose is reducing sugar because it can reduce the Fellings and Tollens reagent. Maltose is also a reducing sugar because it can reduce. Now, what are they made up of? So, sucrose is made up of alpha D glucose and beta D fructose. You can see here glucose and fructose, and the connections are C1, C2, glycosidic linkage. This you can just write down some in the notebook also, like it's made up of glucose and fructose. And how are the glucose and fructose connected? The carbon number 1 of glucose is connected to carbon number 2 of the fructose. The connection between the two monosaccharides is called as glycosidic connection, that is glycosidic linkage. So here we have beta galactose plus beta glucose. So beta galactose is going to be non-reducing and glucose is going to be reducing. So definitely this is also going to be reducing. So either of the two components should be reducing. How are you connecting them? You're connecting them by C1, C4 glycosidic linkage. So here we have C1, C2, here we have C1, C4. When you talk about maltose, it is having alpha glucose and alpha glucose, both are alpha glucose, and the connections are same C1, C4 glycosidic linkage. The 3 examples over here. In the first one, sucrose, it is non-reducing. Second one is reducing. Maltose is also reducing. So that's important for your board practicals also. Now polysaccharides are the carbohydrates which upon hydrolysis yield large number of monosaccharides. Like the formula is C6H10O5, n number of times, where n is a very big number starting from 100 to 3000, which means that you can see that if you just take any carbohydrate and boil it with water at high temperature, pressure and acid. So the acid is mostly like H2SO4 you can add, or hydrochloric acid you can add in this few drops. So you'll be getting large number of glucose molecules. So for board examination, this much is sufficient for you, that polysaccharides, whatever they are, if you boil them in the presence of catalyst like acid, you will get thousands and thousands of monosaccharides like glucose. Okay, so this is the first thing. Now it's not necessary that they are all going to give you glucose, they can give you some other molecules also. The commercial preparation of glucose also we are going to see. This is the commercial preparation of glucose, what we are seeing here. And we've already discussed on the basis of taste, they can be called as sugars or non-sugars. Sugars like your glucose, it's sweet, water soluble, it's very shiny, therefore it's called crystalline. And examples, all the monosaccharides are going to be called as sugars. In non-sugars, we will say that they do not have any taste, they are not going to be soluble in water, they are not going to be shiny, that is they are not showing the crystalline nature, they are going to be amorphous in nature, and they are examples is all the polysaccharides. The third classification, children, on the basis of reducing capacities, which we've already discussed. The carbohydrates can be reducing or non-reducing. You can see this is the way we're going to write the answers in the exam. Reducing can reduce Tollens reagent, Tollens T should be capital, and Fellings reagent, and non-reducing carbohydrates cannot reduce Tollens and Fellings reagent. Second point, they contain the groups like aldehydic and ketonic, which are in the free form, that is they are not involved in any connection or any glycosidic bond. Okay, so if these functional groups, aldehydes or ketones, are involved in the glycosidic bond, they are not free, then the sugar will not be reducing in nature. So here the non-reducing sugars, they do not contain the aldehydic or ketonic group in free form, that is they are involved in glycosidic bond. Examples, all the monosaccharides are reducing in nature. There are few disaccharides like galactose and lactose which are also non-reducing as we discussed, and all the polysaccharides are going to be non-reducing, like for example starch, for example cellulose, and one disaccharide also I have written here which is non-reducing. This is important for your practical examination. Sucrose is non-reducing. The reason for that is the aldehydic and the ketonic group are involved in the glycosidic linkages. Children, how do we prepare glucose and the remaining part of it we are going to see in the next video. So till then, start studying. Your board examinations are coming. If you have any particular chapter which you want that I should take or cover up a particular topic, please write in the comment section. Thank you so much.